Psych 9A, PSB 11A. What can I say? Well, we're on Chapter 3 today. And we had a long weekend. And my first question is, did anybody have a chance to peek at this chapter? Oh, very good. Good. So some of the material in lecture today should resemble what you've seen in the chapter. Has anybody noticed that we're actually following the textbook fairly closely? Yes. I'm following the textbook fairly closely. It's such an expensive thing, we might as well use it. We're now going to begin Chapter 3. And today we're talking mostly about things that you would find in a neurobiology class. And the things we'll be talking about include, well, considering the organism as a machine, or thinking about the mechanisms responsible for certain aspects of our behavior and the behavior of other organisms. Building blocks of the nervous system, important components. Communication among neurons, very important, we'll consider that. And finally, we'll talk about communication through the bloodstream via hormones and things like that. Will we get through the entire thing today? I hope so. If not, well, there's plenty of time to get through it on Thursday. So let me continue. Sort of mechanistic viewpoint of brain and behavior dates back quite a ways, but people point out René Descartes. This is the same guy who came up with Cartesian axes, you know, your x and y axes for mathematics. Same guy. He said, well, let's consider an organism as a machine, and it's machine in the sense that, well, if we put our hand or our foot in a place that's way too hot, we will very rapidly withdraw the limb. And he thought about this a lot and said, energy from the outside, that hot stimulus, is reflected by the nervous system back to an organism's muscles. And so he said, we have reflexes that help us react rapidly to noxious stimuli and thereby prevent damage. So he said, well, in this case, we have a stimulus that excites a sense organ, and we will see that these are heat and pain receptors in the skin. These have axons, which are tiny little cables, microscopic in width, that transmit excitation up to the spinal cord or to the brain. And neurons in the spinal cord or brain then relay messages, excitation back down to a muscle or to a gland, and this can produce action. So if we have a noxious heat stimulus, somehow this signal will be magically transmitted in this diagram up to the brain, but certainly up to the spinal cord. It then gets reflected back down to muscles that will help this person withdraw the leg from the heat. So Descartes says the ability to choose different actions in response to a stimulus depends on the activity of the soul. And this sounds perhaps a little bit primitive, speaking as a cognitive scientist. We would say, well, we know all sorts of things that the brain is able to manage without relying on the notion of the soul. We can think about learning and memory and vision and audition and all sorts of motor activities. Well, we have a whole textbook full of things that help to explain behavior through the brain without relying on the soul. But on the other hand, we don't have complete explanations of everything yet, so we'll leave that word there. Modern neuroscience does indeed seek more mechanistic accounts, however. Nervous system has a whole bunch of neurons in it. The basic unit of communication is a neuron with dendrites, a cell body, and an axon. And effectively, information moves along the neuron in a directed fashion. Information arrives at a neuron's dendrites. Occasionally it's cell body, but there's a lot of information received by a neuron's dendrites. This is neuron A up top here. That information is processed or summed by the cell body, which is this yellow thing up here. And effectively, the cell body is integrating the information received by the various dendrites. If circumstances are appropriate, then what will happen is that a neural impulse, a signal, will be sent down the length of the axon of this neuron. That's effectively its cable that helps it send signals to other neurons. And so if there is a signal propagating along this axon, if there is a neural impulse traveling down, it will arrive at axon terminals, and these are sort of the endings of the axon that then contact the dendrites of neuron B to continue the transmission of information. So neurons have dendrites, a cell body, and an axon, and receive much of their information in their dendrites, integrate that information in their cell bodies, and if that information is adequate, then, well, neural impulses will be sent to further neurons. Our nervous systems have on the order of 100 billion neurons. That's a lot of neurons. The connections among neurons are also fairly nasty because a lot of these neurons are talking to one another. So if you have 100 billion neurons and they're talking to many other neurons, then wow, the connectivity in the brain among the neurons is just immensely fantastic. So there are many kinds of neurons. And you can say, well, there are some neurons that are devoted to sensing stimuli from the outside world. So we have neurons that help us react to a light stimulus. Other neurons helping us receive sound wave information. These are sensory neurons helping us sense things. Motoneurons are neurons that act in a way to guide the movements of muscles in our body. So moto is motor in this case, and the motor system is referring to, well, all of our muscles and auxiliary things that help us move around. Finally, there are neurons that are neither sensory nor motor. They, in fact, lie in the chain of processing between these two extremes, and they're called interneurons. And in fact, most of the neurons in the brain are interneurons. Here's some drawings of several neuron types in the brain. So A, which we'll look at in a little bit more detail, is a black and white drawing of a Golgi-stained Purkinje cell in the cerebellum. And you can see this elaborate dendritic arbor, just like a tree, sort of. You know, it's gathering its information from other neurons. Here's that cell body, and the axon, it turns out, is so thin it doesn't really register in this drawing. Here is an alpha motoneuron in the spinal cord, which with a very widely different shape, and again, a wide arborization in its dendritic tree. There is a spiny neuron. This is now in the brain, in the striatum, and we have a cell body with dendrites. Finally, we have an axonless interneuron in the locus nervus system, and it says axonless. This is not the axon. I think that's the cell body in this case, and that is a dendritic arborization. The whole point is there's millions and jillions of different types of neurons, and it's actually pretty complicated. Do people understand what all these neurons are doing? No, but they would certainly like to, and that's sort of the whole game. There are different ways of visualizing the structure of neurons and cells in the brain. First of all, let me remind you that if we had our skulls removed, this very unpretty thing would be visible, and that would be a human brain. And this is the cortex up here, and this is the cerebellum down here. And what we're doing is looking at different, we're looking at the cerebellum in some detail. So if we were to stain the cerebellum, I believe, with a Nissl stain here, we would find, you see these little ridges along the back of the cerebellum? It has that sort of wrinkly thing going there. So these are the wrinkles in this one on the top right. There's a wrinkle, there's a wrinkle. We go over here, there'd be another wrinkle. And what we find if we look at the neurons that are there, well, we see a layer. Here's a layer, this pink layer. Then there seems to be these large specks on the boundary of this pink layer, which is known as the molecular layer, and this underlying layer, which is now a little bit more purple, the granular cell layer. Finally, we have this thing that looks like the root of a tooth, like we're a dentist or something, and that's white matter. This is actually a collection of axons leading away from the neurons here. So it turns out that those larger spotted cells on the boundary of the molecular layer and the granular cell layer are Purkinje cells, and here they are in beautiful fluorescent micrography, and you can see their cell bodies quite clearly and their nuclei and some aspects of their dendritic arborization. So this would be the molecular layer, this would be the granule cell layer, and this is a whole bunch of Purkinje cells there. Finally, this is sort of a more classic preparation, Golgi stain, that shows pretty clearly the cell body and the arborization. And this was used in the historic days of neuroscience, 100 years ago, to make those drawings that you saw on the previous slide. So let's see. How many people looked at the New York Times today? Well, allow me to look at it for you then. This slide I put in just this morning because here we have something that lies on the intersection of what we talked about on Thursday, which is genetics, and what we're talking about today, which is neurobiology and the visualization of brain structure. So you'll recall that females will have two X chromosomes, and a male will have an X and a Y. So what happens, and this has been known since the 50s, or at least suspected, is that in females there are two X chromosomes. One of them gets deactivated. And we talked about epigenetic factors that help regulate the expression of DNA. Well, it turns out if there are two X chromosomes in each cell, one of them is going to get turned off. And there is a research team that has now succeeded in visualizing which of the X chromosomes gets turned off, the one from the father or the one from the mother. And in this particular picture here, one of the X's getting turned off is indicated by red, and you can see it's generally redder on the left half of the picture, and the other one getting turned off is indicated by green. And so what this is showing is that in the mouse, in the female mouse brain, there's a left to right pattern in the silencing of the X chromosome. And this is a remarkably nice picture showing something really cool, I think. And that's why I stuck it in this morning. If you want to see the actual article from the New York Times, there it is. And you will find it in a journal article. So anyway, that's a nice picture. It's important to visualize how things are laid out. And people are getting ever more sophisticated at that. Now, we already talked about reflexes. Let's look at them in more detail. We had that poor naked youngster burning its foot. How did that work out? Not too well. Well, how does it work if we're now doing something with our hands? Well, there are sensory receptors in the skin. And these sensory receptors will help us respond to pressure. They'll help us respond to increased heat or increased cold. They'll even help us respond fairly directly to pain and noxious stimuli. So there are different sorts of receptors. There are also the hair follicles and things like that. But I'm concerned about these sensory receptors, and the most active elements typically are dendrites receiving information concerning heat, cold, pressure, what have you. This information collected by the dendrites is passed to cell bodies that lie just by the spinal cord, and these have axons that then travel into the spinal column itself, contact an interneuron in the shortest reflex arc that then stimulates a motoneuron. And this motoneuron tells muscle fibers to contract. And that is a very simple reflex arc. So we have the sensing of some event in the skin, and if it is the right sort, that will immediately cause this interneuron in the spinal column to tell a motoneuron to contract the appropriate muscles so that we can pull our hand away or something like that. Now, there's two good terms here, the sort of things that do appear on exams. Well, afferent. The AD, or A prefix, of course, means to or towards. So in this case, we're talking about things moving towards the central nervous system. The abbreviation you sometimes see for central nervous system is CNS. What do you think the abbreviation for peripheral nervous system is? PNS. Okay, now, information from these dendrites of the sensory neurons is traveling towards the central nervous system. In fact, it's only making it to the spinal column, but even so, we would say these are afferent fibers, the ones in red here. Now, efferent, the prefix there is EX, out of, exiting. So now we're talking about fibers that are sending information away from or out of the central nervous system. And there we go. This is exiting, moving towards a muscle to tell that muscle to contract. So those are efferent fibers. Now it turns out that many simple reflexes rely on circuits in the spine, and the brain doesn't really get involved. And the reason for this is you want the reflex activity to be as rapid as possible. You don't want to have information being sent all the way up to the brain and then having somebody think about it and then say, oh, I better pull my arm out of the fire. Well, it's going to take too long. You better just have an automatic reflex at the spinal cord level that retracts the burning finger or what have you. So it's important to point out that most neurons are neither sensory neurons nor motor neurons, even though those are perhaps the neurons with some of the clearest function. Most neurons are interneurons, and it's sometimes a game to figure out what it is they are doing, really hard. The nervous system, to complicate matters, 50% of the cells there are not even neurons. They're called glial cells, G-L-I-A-L, glial cells. They're also called glia, and they are the glue that helps hold the nervous system together, and that's where that word comes from. So these cells have many functions during development and in supporting the function of the mature nervous system, and I'll return to a couple of points in a moment, but let me say now that there are two major varieties of glial cells that you'll encounter. One sort is called an oligodendrocyte, and these produce myelin sheaths that will help facilitate conduction of electrochemical signals along a neuron's axon. And we'll go into gory detail on that in a moment. Then there's a whole different kind of glial cell called an astrocyte, and these do things like mop up excess neurotransmitter molecules and help to meet neuron metabolic needs. Now, has anybody ever heard of functional magnetic resonance imaging, fMRI? Yeah, the idea there is to try to record the use of oxygenated blood by the brain. The idea is when the brain is working hard, it needs more blood to feed it oxygen. If various areas of the brain are especially working hard because of some particular task, well, they'll need more blood. So an fMRI relies on a scanner. We'll probably see this in this coming Thursday's talk, on a scanner that will measure the blood oxygenation level as a function of position in the brain. It turns out that the blood oxygenation level in different regions of the brain changes fairly slowly as time passes, and it's thought to reflect pretty clearly the activity of glial cells, which are regulating the amount of oxygen being taken up by various areas of the brain. So if you're doing fMRI studies, you are looking fairly directly at glial cell activity. You're looking rather indirectly at neuronal activity. It's the neurons that need the oxygen, but it's the glial cells who are likely regulating oxygen flow. Here we are in the spinal cord. Here is a nice cell body of a motor neuron. And you can see its nucleus. When you see a bunch of purple-stained things and you're doing neurobiology and neuroanatomy, what kind of stain is that? I think it's a Nissl stain, N-I-S-S-L, after the person who first popularized it. So this is Nissl stain, and it shows very clearly the sort of purple that you'd expect for the cell body. You also see the dendrites of this motor neuron and various other structures. But I guess the important thing is there are these tiny little guys, and these are the nuclei of glial cells showing up in this Nissl-stained section here. Glial cells are all over the place. They're 50% of the brain. If you now look in the eye, at the back of the eye, you'll find the retina, which is where we have cells that absorb light and turn it into neural information. You'll also find glial cells, and the glial cells in the retina that are easiest to study are the Miller cells, and these are cells that effectively span the entire width of the retina. So they're very prominent structures in that organ. So oligodendrocytes are glial cells that provide myelin sheaths for neuron axons. Oligodendrocytes are effectively creating this fatty membrane that wraps the narrow axon of a neuron. And so if we look at part B here, here's our axon from the neuron. And normally it'd just be this sort of yellow tube, but what the neighboring oligodendrocytes will do is effectively wrap over a period of time these fatty layers around the axon. So in some sense it becomes an insulated cable rather than an uninsulated cable. It turns out that the sheathing provides faster transmission of information along the axon's length. And we'll have a slide shortly that goes into a little bit more detail. But that's the idea. Oligodendrocytes are helping protect axons and provide faster transmission of information. Here is a nice picture showing the many layers of effectively fatty material that are surrounding axons provided by these oligodendrocytes. Now, in multiple sclerosis, it turns out that the primary thing that people can find, the matter is that the axons that should be myelinated are no longer myelinated. So that's the main thing in multiple sclerosis, demyelination, so that these axons no longer have their oligodendrocyte sheaths. And this brings us to an important distinction. There are many areas of the brain where all sorts of information processing is going on, and all the neurons are doing very important things and signaling each other crazily. And you'll find lots of cell bodies. But then there are regions of the brain, and they're shown in these sort of whiter areas here, that are primarily axons. And this area here that I'm pointing to that's whiter, well, that's called white matter. And the reason it's white is because it's very fatty. And the reason it's very fatty is you have a bunch of axons that are wrapped by these myelin sheaths, which is effectively a fatty wrapping. So white matter is effectively a whole bunch of axons going from one place to the next and comprising myelinated axons. What's this stuff called? Do people know the name of this stuff up here that is not white? It's called gray matter. And when you look at it, it's not gray. It's more pinkish or salmonish or whatever your idea of that color is. It's not really gray, but it is called gray matter. And that's where you'll find the cell bodies, dendrites, and unmyelinated axons. And at this point, you have to ask yourself, well, it looks like we're cutting apart brains now. What next? Well, if you're cutting apart brains, it really behooves you to know where you're doing the cuts. And the first thing to know behind that is what kind of cut you get. And let me move to the next one. There are three basic planes that you can make a brain slice with. And I've tried to illustrate them here. The one we saw before in the previous slide is known as a coronal plane. This is a coronal section. And I don't know, if you need to sort of visualize like a crown across the top of somebody's head, or maybe a tiara, whatever the case is, it's sort of slicing, ouch. Okay, so that is a coronal slice. But there are two other possible planes along which one can slice in three-dimensional space. And let's say I now try to separate the left half of my body from the right half of my body with a guillotine coming straight down like that. Well, that would hurt. Let's not do that. But that would be a sagittal slice, separating things on the left from things on the right, like so. Finally, if you take sections of the brain like this, from the front to the back, well, that is going to give you an axial section, and that's illustrated here. So in this particular diagram, these are the eyeballs, and you can see the eyeballs in this axial plane in the front, and we go to the back. So yes, that's the axial section. And of course, an axial section doesn't need to go through the eyeballs. You could take an axial section that's a little bit higher or a little bit lower, just as long as it's going sort of horizontally through the head. Likewise, a sagittal section doesn't need to be straight down the middle of the head. It could be a little bit over to the left or a little bit over to the right, etc. So that with these planes, we not only have an orientation in space, we also have a particular position. Could be left to right, could be up and down, could be back and forth. So in this previous slide, it looks as though we have a coronal section in midbrain. Some ventricles are clearly evident in this section, as is the corpus callosum, and we'll talk about some of these things as the class progresses. So, moving on from glial cells for the moment, let's now talk about how it is that neurons communicate. Well, we know that there are axons somehow sending information to their axon terminals, and the axon terminals connect dendrites of neurons, the next neurons in the information processing chain, or perhaps they're connected to muscle fibers. How do these various pieces of information get sent from one to the next? Well, there are several things we need to know about. First of these is the resting potential, and you have to now think microscopically. A neuron has, of course, it's a cell, so there's all the stuff on the inside of that cell, but then there is a cell membrane, which effectively separates the inside of the cell from all the extracellular stuff. So you can think of this membrane, the cell membrane, as sort of its skin, although it's far thinner. It's a very thin thing with two layers. When the neuron's membrane is stable and it's not changing, then there's usually an excess of positively charged ions in the extracellular space. And that results in a voltage measurement saying that the neuron on its inside is negative. Effectively, the extracellular space has more positive ions, so everything outside is positive. That makes the neuron look negatively charged as a result. When the membrane is stimulated, and again, things are very active in neuronal signaling, turns out ion channels, which rest in the neuronal membrane, open up. And this causes something called an action potential. It turns out that all of the positively charged ions in the extracellular space that are able to fit in these ion channels will effectively move through the ion channels from the extracellular space into the intracellular space inside the neurons. So all the positive charges, sodium ions in particular, flow through ion channels in the neuron's membrane into the middle of the cell, intracellular. And what this means is that, well, there's now a whole bunch of positive ions inside the neuron. And so the neuron now looks like it's positively charged rather than negatively charged. So we opened up the ion channels. The neuron now looks like it's positively charged. But more ion channels get opened that reverse this. In particular, we turn off the ion channels for sodium, we turn on the ion channels for potassium, and the positive voltage that we measured transiently becomes a negative voltage. And it turns out that this excitation spreads physically across space. It leads to propagation of an action potential along the axon. And yes, this is normally the sort of thing you'll see covered in several lectures in a course on neurobiology. In this course, we'll attempt to go over the material in 15 minutes. So if it seems like a lot, it is a lot. There are courses that talk about just this stuff for weeks and weeks. In any event, let me see if I can give you some more details. Here we have our notional axon. Here's our cell body. Here are our dendrites and the axon. And the idea is that the axon is somehow sending signals to further neurons, and we want to know how this happens. So it turns out we have electrochemical events. And we can measure what's going on electrically by using a voltage meter, say. And we can compare the amount of charge on the left to the amount of charge on the right, and we can find out, aha, if we were to measure the resting axon, not doing anything in particular, we would have a negative charge of about minus 70 millivolts. Sometimes it's minus 60 millivolts, sometimes it's minus 80, but you know, typically you're going to find something like minus 70 millivolts. Now, if you were to buy a battery at a store, what might the number of volts be? Well, there's a 9-volt battery. Everybody's seen a 9-volt battery, I guess. This is a lot less than 9 volts. This is 70 millivolts. Now, milli is the prefix for 1,000th, so we have 70 1,000ths of a volt. So this is actually an electric potential that is far weaker than you'd find in your normal 9-volt battery. But it's still appreciable and measurable once you have the proper equipment. So what happens if you now measure this electric potential as time passes? Well, that's this horizontal axis here, marked in milliseconds. A millisecond is 1,000th of a second, so this is pretty quick here. What we find, oh, what just happened? Oh, that was interesting. I wonder what I just did. Very exciting. Good. There it is. Too many buttons. What happens is that minus 70 millivolts, the resting potential, well, transiently, that's no longer accurate. You get this positive spike with a very quick return to a negative potential. And over the course of several milliseconds, you're back to the standard resting potential. This is known as an action potential, and it turns out that this action potential moves down the length of the axon. So this action potential is shown as a function of time here in this thing here, and what we've got is a resting potential of minus 70 millivolts, and something happens to this neuron that makes it want to send a message to the next neuron. So in fact, it has been activated above its threshold level, and that is a threshold for firing an action potential. If the cell membrane right at the base of the axon here exceeds the threshold level, then an action potential will be sent, and we have this momentary depolarization, increase in electric polarization. We still have more slides. You thought this was going to be quick. So sorry. Resting potential. It turns out that if we have threshold reached because of, say, the signals that we've collected in our dendrites, we will generate an action potential. And what happens is that the sodium channels in the membrane will open up. And the basic idea is that there's lots and lots of sodium ions in the extracellular space. Sodium ions are positively charged. So we have all these positively charged ions in the extracellular space. We do not have them in the intracellular space. And that's why the neuron looks like it has a negative potential. The sodium channel's open in the membrane. It's effectively opening the floodgates. Sodium ions now flood into the neuron. And what happens is that all of those positive charged ions will give the neuron now a positive potential. There'll be this very rapid depolarization, which can reach, say, positive 50 millivolts. Now, as soon as those sodium ions have flooded in, well, the channels get closed once again, no more sodium messing around. And it turns out that there are further ions, such as potassium, that have their own channels, and these ions will flow across the membrane to bring the potential back down to this negative value. And in fact, there's a period here shown as the refractory period between 2 to 4 milliseconds where this neuron will be unable to send another action potential. No matter how much we're telling this one neuron that we would like it to send an action potential, well, if we're telling it during the refractory period, too bad. It's unable to generate an action potential during that period of time. So there's some things here. The sodium ions rush into the cell because the channels open, but there are other things that are not mediated by channels, but rather by pumps. And what happens to all of that sodium that you just let into the cell? Well, it has to be pumped back out. And so there are pumps in the cell membrane in addition to channels. The sodiums flow in very rapidly through the channels, but then it gets pumped out so that we can send another action potential several milliseconds later. So there are these pumps. And it's really pumps that help get us back to this negative electric potential. Depolarization means we have a change potential that makes it more positive. Hyperpolarization means we have a decrease in electric potential that makes it more negative in value. So that this would be a depolarization in this ascending limb of the action potential. This would be a depolarization. They say repolarization here. Hyperpolarization in this stage here. Good. So I see a hand.
About the last slide, how
Yes.
depolarization doesn't occur at the same place as repolarization?
Same place.
Like the same point on the graph?
Well, what happens is that we've got a sequence of events, and it's shown a little bit here, and in fact you'd need a proper textbook in several weeks to see all the events, but the primary ones again are the opening of the sodium channels, which causes this increase in the potential. But as soon as you get here, effectively, well, the sodium channels close up, no more sodium allowed into that neuron, and then you have to ask, well, how do we get from that positive potential back down to the negative potential? There are potassium channels that open, there are also chloride channels that open, and there are also pumps that start working. And the effect of all of these mechanisms in the neuron cell membrane is to get those positively charged ions back out into the extracellular space. And that takes time. So if you look at some one point along an axon, you will see these events take place as a function of time, say 5 milliseconds, what have you. It turns out, and I'll show this I think I show it in a slide shortly. No, I don't. That this propagates from one location in the axon to the next. And so that the opening of the sodium channels will happen here, then a millisecond later it will happen here, and a millisecond later it will happen here, and eventually this action potential will get all the way to the axon terminals where the next neuron is contacted. So that we not only have an action potential thought of as a function of time, it also propagates in space down the length of the action potential. I'm surprised there aren't more questions, in fact. This is really hard material. I see a hand. Good.
With changing the replication of the, let's say, the fork or whatever, where the neuron starts, like the propagation starts, like the starter, would that change the direction where it propagates?
Well, let me answer that in two ways. There's a thing called an axon hillock. And oh, you know the answer to the question then. Okay, well, right at the start of the axon, at sort of the base of the soma, you'll find the axon hillock. And it is there where an action potential will start propagating down the length of the axon if the threshold polarization is reached. And so the normal activity effectively means, well, we're looking right here to see if the dendrites are giving us enough stimulation. If they are, well, we'll send an action potential down the length of the axon. Can we send these signals in both directions, in effect? Well, we can certainly send them in any direction that we wish. They are bidirectional, is the answer. But they don't normally operate that way. In normal action, you have an axon hillock, and at that point is where you find potential starting and then traveling down the length of the axon, eventually to their axon terminals. So in normal function, axons are unidirectional. Okay. Propagation is the term whereby we have the action potential moving down the length of the axon. And it turns out that propagation is faster if the axon is myelinated. And it turns out that if you measure depolarization, the action potential waveform, you'll find that it effectively skips from one node of Ranvier to the next. And I think I got some of those guys, nodes of Ranvier. There they are. Here's a little sausage. Here's another little sausage. Here's a little sausage. These are the sausages formed by the myelin sheaths of the individual oligodendrocytes. In between these guys are the nodes of Ranvier, and what happens is that the electrical activity, this electrical chemical activity will jump from one node to the next, spending minimal time propagating in these areas that have been sheathed. So in fact, myelination will make action potential propagation much faster because of the jumping of the action potential from one node of Ranvier to the next. Let me go back. Here we go. Saltatory conduction is the name for that. And saltatory basically means jumping. Anything that's saltatory is jumping around. So we're jumping from one node of Ranvier to the next with a myelinated axon. Now, it turns out that an action potential obeys an all or none law as follows. Once the action potential is initiated, once it is launched at the axon hillock, at the base of the cell body, further increases in stimulus intensity have no effect on its magnitude. So the electrical shape of an action potential for a particular neuron is always the same. It might start at minus 70 millivolts, get up to positive 50 millivolts, and then come back down over the course of, say, 5 milliseconds. The action potential for that cell will always be the same thing. no matter whether we're looking at a bright light or a dim light or what have you. The action potential is all or none. Once it happens, it looks the same. And here's the problem then. How can stimulus intensity be coded if all action potentials are the same? And the answer, or there's a whole bunch of answers, but here are some of the basic ones. Hey, if you have a more intense stimulus, you can have your neuron fire more action potentials. And that can be used as a signal for intensity. So the more intense the stimulus, the more frequent the action potentials. And what people talk about is the firing rate of a neuron. And what they're referring to is how rapidly the neuron is sending action potentials down its axon to further neurons in the information processing chain. So here we have in the plots these functions of time. So that's our horizontal axis, the passage of time. And what we are actually recording is voltage. This is not right. We're not recording firing frequency here. This is wrong. What we're recording is voltage. And we see spikes of voltage when an action potential occurs. And if we have a very weak stimulus, there may be very few action potentials. And that's shown in the top plot. Now, here we have 160% of threshold intensity. Well, pretend we're talking about lights. And you can have a dim light. You can barely see it, and maybe the neurons are barely responding. You can turn up the intensity of the light. Now you can have effectively a stimulus that exceeds threshold for a particular neuron by some fixed amount. Maybe it's 160% of the threshold intensity. And we now look at the action potentials, boom, 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 boom. This is now firing lots of action potentials. So in the same period of time, more action potentials. We would have to say that the frequency of action potential firing is much higher in this case than in the original case. Finally, we turn up that stimulus way high, we have a very high firing rate. We have a high frequency of response. So does that make sense? We can code for intensity simply by firing more action potentials. Now, there's something in the book that just has to be mentioned. There's this guy, Levy, back in the 1920s, who did some really famous work on mechanisms by which neurons signal. And in this case, he's interested in the neural control of the heart rate. So there's this guy, Levy, doing these experiments. Eventually he gets a Nobel Prize. How many people have seen Pulp Fiction? That's a really gory movie. I don't want to go into details, but somebody receives a shot at one point to revive her. That chemical was effectively discovered by this guy and deciphered as a leading neurotransmitter that affects the contraction of heart muscles. Norepinephrine, to be specific, that will increase heart rate. And that is the Nobel Prize winning work by this guy, Levy. In any event, in this particular thing, we're looking at signals sent by the vagus nerve. And the vagus nerve tells the heart what to do to a certain extent. So let's see what we got. What did he do? He placed two still beating frog hearts in the jars of saline solution. Now, when people say saline solution, what are they basically saying? Salt water. Okay, so we have some salt water. Here is a frog heart with the vagus nerve attached. Here is another frog heart with its vagus nerve attached. Fine. With an electrode, he stimulated the vagus nerve of the heart in jar A. So here's our stimulating electrode. This nerve is now stimulated by all this activity, and something is going on here. Turns out that it slows its beating. So if you stimulate the vagus nerve the way Levy did, then that heart will slow. This is not the norepinephrine experiment, by the way. This heart will slow its rate of beating. And his idea was, well, you know, if this is done electrically, there's no reason to think that there's anything that we can take away from this beaker A that might do something to the heart in beaker B. If the effect of the vagus nerve on the heart A, namely to slow it down, is mechanical, there is no reason to think that anything about beaker A will have an impact on the heart in beaker B. But if what's going on in beaker A is chemical, then perhaps there are chemicals being released into the saline solution that will then have an effect on heart B. And that's exactly what it is. The vagus nerve releases chemicals. They get into the saline solution in the beaker A, where we have this slowed heart. We can pour that saline solution into beaker B, and we will find out that, yes, that heart has slowed its beat. And so, hey, that's the result. The heart in jar B beats slower when fluid is added from jar A. This wouldn't happen if the vagus nerve acted by an electrical signal. It wouldn't happen if it worked by some mechanical means. It would happen, though, if there's something chemical going on and those molecules are released into the saline solution. So, the vagus nerve sends chemical signals to the heart. Chemical signals. Aha. Well, you've probably guessed that we're now going to talk about chemical signals. Neurotransmitter molecules. These are released at axon terminals made with dendrites, and the points of contact are called synapses. A neurotransmitter molecule will cross the synapse from the axon terminal to the dendrite. The molecule will latch onto receptors in the neuron membrane of the postsynaptic cell, and this can trigger response in that further cell. So the first thing, there are these points of contact between presynaptic neurons, which are effectively speaking through their axon terminals, and postsynaptic neurons, which are receiving signals with their dendrites. And these points of contact are the synapses. And these are effectively localized areas where neurotransmitters are used to signal information. So, what we have here are some electron micrographs showing us synapses and vesicles containing neurotransmitter molecules. I always enjoyed seeing pictures of vesicles. So let's see, we are in the hippocampus, and we've seen these hippocampus cells, Purkinje cells, earlier today. You probably wonder where the hippocampus is. Well, what section of the brain is this? Is this coronal? Is it axial? Or is it sagittal? It's one of these sections. And if we take it at the right amount of left and right, well, we can probably get a good slice through the hippocampus, which is shown here in teal, or whatever that color might be. I don't have a lot of color names in my vocabulary, but somehow teal ended up being one of them. Isn't that strange? Okay. So here we have a neuron. Here we have out in its dendrite something that we're going to blow up. See that little rectangle? Here we have blown it up. And what we find is that there is a presynaptic terminal. And if you look very closely, there are little circles. Do you see those little circles here? Those circles are filled with neurotransmitter molecules. The neurotransmitter molecules, the vesicles will move to this border, the membrane of the presynaptic neuron, and they will then release the neurotransmitter molecules into this cleft, the extracellular space between the presynaptic neuron and the postsynaptic neuron. Here is yet another example. Presynaptic vesicles right across from this synapse and the postsynaptic neuron. Here's another nice example here. So, the idea then is that the action potential will propagate down the length of the axon from the cell body to the axon terminals, and there will then be synaptic connections made with further neurons, postsynaptic neurons. And so we have this in cartoon form now. Here's our presynaptic neuron axon terminal. Here are some vesicles filled with little transmitter molecules marked T. These vesicles, when the cell is firing, will move up and release their contents into the synaptic cleft, which is this extracellular space right here where you see the little T's. And right across in the postsynaptic membrane, you will find these guys. These are receptors for particular neurotransmitter molecules. And in this particular example, if we have little molecules shaped like T, well, you might expect a receptor in the postsynaptic membrane to have a place where that T molecule fits well. And if these T molecules fit well into these receptors here, then something changes in the postsynaptic cell. And in this case, well, look, this channel was closed before the T transmitter molecules came, but now they've arrived. They fit into the receptor molecule, and this channel is now open. So effectively, the postsynaptic cell changes in some way because of these transmitter molecules. And this is a good example right here. An ion channel opens as a result of these transmitter molecules. Now, these transmitter molecules are floating around in the synaptic cleft, and you soon say, well, there's a lot of this stuff floating around in the brain. That might pose a problem. But in fact, the transmitter molecules turn out to be deactivated in a number of ways, including being broken down by cleanup enzymes. So enzyme molecules in the synaptic cleft that make these effectively inactive. And then there's also a process of synaptic reuptake that you'll find in both neurons and glial cells. Effectively, these cells will be looking for the transmitters that have been released into the synaptic cleft, and they will reuptake these molecules so that their effects are of short duration rather than of long duration. So what are some of these molecules? Well, acetylcholine is released at many synapses, and it's also the main transmitter between nerves and muscles. Effectively, the release of acetylcholine makes muscle fibers contract. So that's an important one. Serotonin is another involved in many of the mechanisms of sleep, mood, and arousal. Gamma-aminobutyric acid, or GABA, is the most widely distributed inhibitory neurotransmitter. And now I have to point out one thing. An excitatory neurotransmitter will cause the postsynaptic neuron to depolarize. And one of the primary mechanisms is shown in that previous slide. If we open sodium ion channels, then the sodium will rush into the cell, and that will cause a depolarization, an increase in positive electric potential. Now, it's also possible for a neurotransmitter molecule to impact channels that will make the cell on the inside more negative. So that turns out to be an inhibitory neurotransmitter molecule when the main impact is to hyperpolarize the postsynaptic neuron. So GABA, it's an inhibitory transmitter. Glutamate, an excitatory transmitter, is all over. Plays a crucial role in learning and memory. Norepinephrine controls arousal level, influences wakefulness, learning, and memory. Finally, dopamine makes people dopey. No, that's not true. It influences movement, motivation, and emotion. Implicated in Parkinson's disease, for example. Now let's see. Third point on this slide, we looked at the first two. We have cleanup enzymes, and we have reuptake. Now, the very important lock and key model says transmitter molecules will affect the postsynaptic membrane only if the molecule shape fits certain postsynaptic receptor molecules. So when I said those little T's had to fit into the proper place in the receptor molecule, I was not making that up. That's effectively the lock and key model. Postsynaptic receptor molecules effectively have to be contacted the proper way for a transmitter molecule to have any effect. These pink guys want T's. These blue guys want angled T's. I guess those are tacks or whatever they are. Anyway, they have different shapes. So they're selective for different transmitters. That's the lock and key idea. So it turns out that we can influence a neurotransmission of information in a number of ways. And there are two primary categories of influences. One of these influences is shown by agonists, and what an agonist does is to enhance the effect of a neurotransmitter. So an agonist will enhance the effect of a neurotransmitter. The opposite of that then would be an antagonist, which is some way to impede or degrade the effect of a neurotransmitter. So, drugs that are agonists or antagonists can work by, well, blocking the transmitter's synaptic reuptake. So if we were to block the reuptake of a transmitter, we'd have to say that transmitter is now hanging around in the synaptic cleft more than it should be. We would in fact be enhancing the action of that neurotransmitter. So if we had a drug that blocks reuptake, is it an agonist or an antagonist? Agonist. We are enhancing that neurotransmitter's action because we're letting it sit in the synaptic cleft longer than it normally would. So it's hanging around at the synapse more than it should be. So it's going to have a stronger effect.
An SSRI?
Say it again?
An SSRI for depression?
I don't know what an SSRI is.
If there's a shortage, when people are depressed, there's a shortage of serotonin. And so, like,
Oh, yes, SSRI. Yes, exactly correct. Sorry, I didn't know that. We'll see that, I think, on the next slide. If we counteract a cleanup enzyme, we'll have roughly the same effects. We're effectively saying, well, we're not going to break down this neurotransmitter with the efficiency that we normally do. Therefore, this neurotransmitter will sit in a synaptic cleft, perhaps activate the postsynaptic receptor molecules. More frequently, once again, we have an agonist. If we mimic a transmitter's action, that's also an agonistic effect. So here we are. Now we have all sorts of examples of agonists and antagonists. And over on the left, we'll start with the agonists, and these are effectively going to enhance the action of a particular neurotransmitter. Agonist drugs can increase the release of neurotransmitters, and that's the main effect of amphetamine. And let's see, amphetamines are agonists for norepinephrine and dopamine, as it turns out. Turns out agonists can counteract the cleanup enzymes that break down the transmitter. We mentioned that already. We also can block the reuptake of neurotransmitters. That would include Prozac, and where did Prozac go? Aha, Prozac SSRI, blocking the reuptake of serotonin, and cocaine. Where is cocaine? Blocking the reuptake of serotonin, norepinephrine, and dopamine. The main effect of nicotine is to mimic a particular neurotransmitter. So in fact, nicotine mimics acetylcholine, ACH. So if there's acetylcholine transmission of information at a particular synapse, then a nicotine molecule will pretend to be an acetylcholine molecule and get that synapse to operate. So that is an agonistic effect of nicotine. Over on the other side, the right side, we see some effects of antagonists, and these can, well, block the release of neurotransmitters, and that would be a main effect of AMPT. which effectively is blocking the synthesis of tyrosine and catecholamines. We have antagonists that can destroy neurotransmitters at the synapse, and we can also effectively mimic a neurotransmitter and effectively bind to a receptor in a way that causes it to function less well than it would otherwise. So effectively we can block the action of these receptors with properly chosen antagonist drugs. And this is the main effect of propranolol, which is a beta blocker. Blocks the action of, well, effectively mimics the action of norepinephrine and blocks receptor channels. Ditto for Haldol, which is doing the same thing for dopamine. And you can see that there's a whole bunch of science involved in this neural business. And the particular science here is neurochemistry. There's all sorts of neurochemistry and neuropharmacology going on to understand how these drugs work on the transmission of information in the brain. Now we're getting close to the close of this lecture. It turns out that we have neurons releasing chemicals not only across synapses where they have a very, very, very microscopically local action on postsynaptic dendrites, or perhaps muscle fibers. We've also got neurons releasing molecules into the blood. And this can cause all sorts of things to happen throughout the body. So the blood circulation, as you know, brings oxygen and glucose molecules, honey, sugar, to the nutrient-hungry brain. But it also aids communication by carrying hormones secreted by endocrine glands. And so there are a host of endocrine glands in our bodies that release hormones into the brain. And here's a brief listing. The anterior pituitary gland in the middle of the brain is called the body's master gland because it triggers hormone secretion in many of the other glands. So it's sort of the boss telling various endocrine glands what to release and when. Posterior pituitary prevents the loss of water through action on the kidney. The thyroid, well, has anybody ever had a pet or some other beloved creature with like hyperthyroidism? If you're familiar with hyperthyroidism, the pet will shrink and start getting very nervous and run around. It's going to be very, very active and will burn up all of its body energy at a higher rate than average. Thyroid. In any event, there are various things going on in the thyroid that affect metabolic rate. My parents have a cat who had hyperthyroidism. That's what I was thinking about. Islet cells in the pancreas, of course, affect utilization of glucose. Adrenal cortex, a place where you get things that help one respond to stress, with some effects on sexual behavior, and we'll get there in a moment. The adrenal medulla increases the sugar output of the liver, blah, blah, blah. Ah, the ovaries and the testes, finally. The ovaries. One set of hormones, estrogen, produces female sex characteristics and is relevant to sexual behavior. Another, progesterone, prepares the uterus for implantation of the embryo. Testes produces male sex characteristics and is relevant to sexual arousal. And here are locations of these various endocrine glands. So, the endocrine system has much in common with chemical communication between neurons, suggesting that these things have common evolutionary origin. The gland cells releasing chemicals, neurons releasing neurotransmitters. And that's all I have to say today. Thank you for bearing with me.